The morphology and distribution of cells on microcarriers affects their growth, kinetics, and even their productivity. Therefore, microscopic examination is an important practice in microcarrier culture. However, a number of microcarriers, such as glass and polystyrene, are based on materials which are refractile under microscopic examination, thus making the examination of cell morphology and distribution difficult. In such a case, staining with crystal violet at least makes a portion of cells on the bead more visible, especially when multiple layers surround the beads. Another method of improving the observability of cells on these microcarriers is to apply fluorescein diacetate stain. The stained cells appear green when observed under a fluorescent microscope. When fluorescein diacetate is applied together with ethidium bromide, the viable cells appear green, while the dead cells appear orange. Such a staining technique can also be applied to macroporous microcarriers, which are not easily observable in light microscopes. Cells grow both on the external and internal surface of these beads. Using fluorescein diacetate and ethidium bromide stain, the viability and even the cell distribution can be estimated under a fluorescent microscope. It is obvious that such a technique can be readily applied even to conventional microcarrier culture to determine the viability of cells in different growth stages. This videotape is based on the laboratory manual developed by the Biochemical Engineering Laboratory in the Department of Chemical Engineering and Materials Science, University of Minnesota. The demonstrations were performed in the Biochemical Engineering Laboratory and at the Central Research Facility of the Institute for Advanced Studies in Biological Process Technology.